Now, I would like to turn the podium over to my colleague, Dr. Diana Wetmore, who will introduce our Scholar Experience presentations. Dr. Wetmore is Vice President of Therapeutics Development and Director of the Therapeutics Development Center. She is a key ingredient in the secret sauce, if you will, that differentiates Harrington Discovery Institute in the drug discovery space. Welcome, Diana.
Thank you, Seth. It's a real pleasure to be here today and present our Scholar Experience lectures. But before we get started, I'd like to take a minute just to remind the audience of the depth and breadth of capabilities we've put in place to help investigators advance their drug discoveries into licensable packages that attract commercial partners. Our Therapeutic Development Center, or TDC as we refer to it, includes an advisory team of drug development experts and project managers who work as extended members of the Scholars team to move their technology towards the clinic and to use in patients. Our TDC experts have spent their careers in pharma, and they bring diverse skills in preclinical and clinical planning, pharmacology, biology, and biophysics, formulation and chemistry, as well as commercial and business development. In addition to the drug development experts, we have project managers who work with the scholars to develop a project plan that enables them to meet major milestones, utilize their funding most effectively, and maximize the value provided by the TDC advisory team. It's a winning formula we've developed over the years, and for me, it's especially satisfying to hear from scholars such as our next two presenters, who will now share how they've made significant progress in their pursuit of new therapeutics. I'm pleased to introduce our first scholar, Dr. Rosa Bacchetta. Dr. Bacchetta is an associate professor at Stanford University and a member of the Maternal and Child Health Research Institute. She received her MD from the University of Turin in Italy, where she also completed her fellowship in general pediatrics immunology. Her scientific interest has been in the genetic and immunological basis of primary immunodeficiencies that might be treated by gene therapy. As a pediatrician specializing in immunology, Dr. Bacchetta has made it her professional goal to challenge the limits of what are often referred to as inexplicable and untreatable diseases. Her research applies current scientific knowledge to understand the mechanism of the impaired cellular immune function underlying these diseases in order to develop creative therapeutics. She's here to tell us about her Harrington project, which is focused on genetically modified T cells to treat IPEX syndrome, a rare disease inherited in male children.
Good morning, everyone. I really feel honored to have the opportunity to give feedback to the Harrington Discovery world and team about the project of cell and gene therapy with engineered CD4 positive LV-FOXP3 cell product for autoimmune genetic disease called IPEX syndrome. So as probably many of you know, autoimmunity is the consequence of a profound breakdown of the immune tolerance when the immune system start to attack different organ and tissues. And really what we see in the pathology is the tip of the iceberg of a profound alteration of the immune system itself, which is, only partially understood nowadays. And already, I think about 20 years ago, it became clear that autoimmunity can be due sometimes to genetic defect, so to only one gene defect that cause alteration of the immune regulatory part of the immune system and generate the pathology. In the past 20 years, since the first discoveries, there has been really an increase in these genetic diseases of the immune system, and in particular, it has been possible to define much better what a set of this disease called T-regopathies, because the gene that are altered there alter in particular the function of or the development of a set of cells which are called T regulatory cells. The first to be discovered has been the mutation of the gene called FOXP3. And FOXP3 is the cause of IPEX syndrome, that stands for immune dysregulation, polyendocrinopathy, enteropathy, and is an X-linked disease, so only manifested in male. So FOXP3 is important because, the T regulatory cells are very important at the beginning of life. We are born with functional T regulatory cells that immediately are able to control all the other lymphocytes in the body so that they don't attack our own tissue, so prevent autoimmunity. However, in patients with IPEX, these cells come out of the thymus or unable to function, and therefore all the lymphocytes are indeed free of attacking the cells and tissues. The most common manifestation are enteropathy because of the gut aggression. So this patient manifests with very severe diarrhea early on in life, eczema, so they have really eczema like atopic dermatitis all over the body, and sometimes type 1 diabetes. And as you can see, almost half of the patient can have the onset of the disease within the first months of life, and the rest basically before 1 year of life. Nowadays, 330 cases have been reported in literature approximately, and we know that even late onset IPEX or mild IPEX is possible, and so that many other symptoms can indeed occur in this disease. And of course, as pediatrician, we know that a person is a person, no matter how small. And studying rare diseases, I really realized that the disease is a disease that deserves treatment, no matter how rare. And so already several years ago, we have realized that, for this IPEX disease, the current treatment available are far from being ideal. So pharmacological immunosuppression is partially efficacious. And as you can imagine, so to have, pharmacological immunosuppression so early in life can really lead to a lot of side effect, as you can see here. So the outcome is very poor. And bone marrow transplantation or hematopoietic stem cell transplantation can be a cure, however, is again, not ideal, and it's not available to everybody. So we came up with the idea of, taking the cells of these patients and use a gene therapy approach to really modify these cells by putting inside the cells, so inserting a normal FOXP3 gene in a way so that the gene, it is always expressed, so the protein is always there, and the cells that are manipulated as such, they become regulatory cells. And once they become regulatory cells with our in vitro manipulation, they can be reinfused to the patients. And in addition, these cells have a sort of, what we insert is also a sort of flag on the surface, so that we are able to purify the cells while we manipulate them, and also to trace them in vivo. And so these cells, it's very complicated, but I just want to give you the message that they are really becoming as naturally occurring T regulatory cells, as the T regulatory cells that we always have in the body by any means. So their function and the way they look. And so over the past 3 years, we have been really going further with this project and completed the preclinical validation using mainly humanized mice, demonstrating the safety and the efficacy to control autoimmunity in these models. And we have also been able to establish the process, instead of in a small dish in our research lab, in large scale, ready to go into the patients. And we have filed and obtained the approval for the phase 1 study from the FDA in October 2021. So we have now opened the first in human Treg replacement therapy, the way we call it, for IPEX syndrome, and also this cell product obtained the orphan drug designation, and the IPEX was designated as a rare pediatric disease. And so what we hope to obtain by taking the cells of the patient, manipulating them in vitro in a 3 weeks culture and then given back is, of course, to be safely able to control the autoimmunity in these patients. So the journey has been as follows. In 2019, immediately after we had the pre-IND meeting with the FDA, the HDI kicked in with its support, and this has been, as you can see, by the time the project basically didn't have any financial support. But what really the HDI brought to me and to all of us is not only the money, but really the support of expertise in the field that, depending on our needs, really were there, always available to respond to questions. So we had a regular meeting with them, but also by any needs we had, I could just write an email and get all the appropriate setup and answer, etc. So this was really a tremendous values to us, and really started the bridging from the bench to the bedside of the treatment that we last year received and achieved with the CERM grant that decided to support the clinical trial. And of course, the participation of patients and family was amazing all along the way. So right now, this engineered Treg product is therefore listed by the clinical trial gov as a Treg product for the clinic, the only one which is made of engineered T regulatory cells. And of course, we are looking for having this product in IPEX, but also in other indication for more common autoimmune diseases that nowadays only have pharmacological treatment as a potential cure. This has been, of course, possible with the Center for Definitive and Curative Medicine of Stanford, a very strong infrastructure made of Stem Cells Institute and GMP facility, and the hospital, in particularly the pediatric hospital. And of course, with the help of many young investigators and many incredible large number of collaborators. And thank you so much, extremely grateful for the HDI support.
I hope you'll agree that was very inspiring. And now for our second Scholar Experience presentation. Dr. Mark Wine is an assistant professor of medicine at Harvard Medical School and an associate member of the Broad Institute. He received his bachelor's and master's of science degrees from Yale University and his MD and PhD from Harvard Medical School. He was trained in internal medicine and endocrinology at Massachusetts General Hospital, where he currently runs his research laboratory and clinical practice. Dr. Wine's pioneering work focuses on the signaling cascades and gene networks that bone cells use during normal skeletal development and remodeling. His lab's aim is to understand and exploit normal bone regenerative pathways in order to develop new treatments for osteoporosis, fracture repair, and other skeletal diseases. He is here to tell us about his Harrington project focused on creating a new therapeutic for osteoporosis.
Great. I wanted to start by thanking the Harrington Discovery Institute for their generous support over the years and for this opportunity to return to the annual meeting to share an update on the status of our project. I was a scholar innovator from 2018 to 2019, and it's really exciting to be back here today. I'm an endocrinologist with a clinical focus on osteoporosis and metabolic bone diseases. In doing so, I spent a lot of time thinking about parathyroid hormone. To remind you all, parathyroid hormone is produced by the parathyroid glands in the neck, and the job of this hormone is to maintain blood calcium levels in the normal range. If our blood calcium levels drop too low, our heart and our brain does not work perfectly. So it's very important to maintain normal blood calcium levels in a very tight range. So as blood calcium levels start to drop, the parathyroid gland senses that and releases parathyroid hormone. And PTH, or parathyroid hormone, aims to restore normal blood calcium levels through actions both in bone and in kidney. In bone, parathyroid hormone promotes bone destruction because 99% of our calcium in our body is actually stored in the solid phase in bone. In addition, parathyroid hormone works in the kidney to promote the reabsorption of calcium. Today, we're going to be focusing on the actions of parathyroid hormone in bone because of the translational potential of these actions. So now we're zooming in on our bone tissue. When parathyroid hormone is released into the bloodstream, PTH binds to receptors on bone cells, osteocytes, which are cells that are embedded in bone, and osteoclasts, and osteoblasts, I should say, which are the cells that build bone. When PTH binds to its receptors in these cells, it leads to the production of a hormone called RANK-L, or rank ligand. And what rank ligand does is it drives the differentiation of osteoclasts, which are these Pac-Man-like cells that live on bone and promote bone destruction. So PTH binds to receptors on osteocytes and osteoblasts, leads to the production of rank ligand, which in turn stimulates calcium release. If this were the only thing that PTH did, our bones would constantly thin and we would lose bone mass very quickly due to blips in parathyroid hormone. So this is a smart hormone and it prepares for the future. Normally, osteocytes make a protein called sclerostin, which slows down bone production by osteoblasts. PTH lowers the expression of sclerostin. So in doing so, PTH both stimulates bone resorption by osteoclasts to promote calcium release, but also promotes bone formation by osteoblasts. And this leads to a bit of a paradox in the effects of parathyroid hormone on the skeleton. When PTH levels are always high, the osteoclast effects win out and we lose bone mass. However, when PTH levels are intermittently high, this actually preferentially triggers bone formation with important implications for osteoporosis. Osteoporosis is a major problem in our aging population with high associated morbidity, as is observed here by progressive height loss and due to compression fractures, and mortality. The 1-year mortality after a hip fracture due to osteoporosis is up to 20%. Osteoporosis is due to a loss of bone mass. As you can see here, thin bone is weak and more likely to fracture. And this is really due to an imbalance between bone destruction by osteoclasts and bone formation by osteoblasts. This is a major public health problem, and efforts have gone in to develop a number of FDA-approved medications for osteoporosis, which are listed here. The majority of our currently used osteoporosis therapies actually block bone destruction by osteoclasts. And this is a kind of a short-term strategy that can be somewhat effective, but certainly does not cure disease. Analogs of parathyroid hormone are really the main bone anabolic therapies that are used, but these are once daily injections, which is really a major barrier for our patients with chronic asymptomatic diseases. So I think everybody in the field would agree that a major unmet need here is the development of orally available bone anabolic agents. So the goal of our laboratory is to understand how PTH works in bone cells in order to inform the development of new therapeutic strategies to treat osteoporosis. Motivated by those efforts, work over a number of years now has defined a signaling pathway used by PTH in target cells in bone to stimulate bone formation. So salt-inducible kinases, isoforms 2 and 3, are normally present in the active state in bone cells, and this leads to phosphorylation of substrates of these kinases and the retention of these proteins in the cytoplasm. When parathyroid hormone binds to its receptor through a signaling cascade that involves protein kinase A, salt-inducible kinases are inhibited, and this leads to the reduction in the phosphorylation of the substrates of these kinases. So these proteins then move into the nucleus and regulate the expression of target genes that in turn regulate bone formation. So because PTH signaling inhibits salt-inducible kinases, a prediction of this model would be that direct small molecule inhibitors of salt-inducible kinases would show effects that are similar to those of PTH. So before starting the Harrington Discovery Institute collaboration, we had characterized a compound called YKL-05099, which is a first generation SYK inhibitor. This compound has expected effects. It lowers the phosphorylation of SIK substrates, leads to changes in target gene expression, and has effects on bone in vivo. However, this is really a non-specific inhibitor with many toxicities that are related to the off-target actions of this compound. So the high-level goal of our collaboration with the Harrington Discovery Institute was to develop pharmaceutically acceptable, orally available compounds that inhibit SYK2 and SYK3 to stimulate bone formation. And this is really where the Therapeutics Development Center comes in. It was really such a great pleasure to be able to work with Bob, Bill, Pat, and Jan, and very importantly, to have kind of Mike Hallen there every step along the way to ensure that the project was moving forward. So at a very high level, what we did was to begin with a homology model of SYK2, which is one of the two kinases that we wanted to target. And this was developed in collaboration with John Culp at Conifer Point. We performed virtual docking for compounds that might bind the active site of this target. And identified a hit chemotype for iterative medicinal chemistry. And here you can see with design redesign cycles, our compounds became more and more potent. We assessed compounds for activity, selectivity, activity in cell-based systems, selectivity, and importantly, compounds with drug-like features. And from all of these efforts, we ended up with a lead compound called SK124. So how did we do? This is a dendrogram, a picture of the majority of the kinases that we have. And when we test YKL-05099, our initial kind of tool compound, we see that this compound targets actually 43 of the 300 kinases in these kinome profiling assays, including SYK2, which are the SYK2 and SYK3, which are the red dots at the bottom of the screen. In contrast, SK124 looks a lot better. Instead of targeting 43 kinases, there are only 7 off-target effects. And this compound has very potent activity, both in vitro kinase assays and cell-based target engagement assays, as is shown here. So after the Harrington Discovery Institute collaboration wrapped up, we teamed up with Radius Health to test the in vivo effects of this compound. We administered it to mice once daily for 3 weeks at 3 different doses, and they were able to observe measurable amounts of the compound in serum. And importantly, we see increases in bone mass, as is observed here, looking at a micro CT image. In the vehicle-treated mice, there's very little bone, while while in the PTH, which is a positive control, and SK124 treated mice, we can see an increase in bone volume. And in addition, we see that this is due to increased bone formation, as measured here by these dual calcine demeclocycline labeling experiments. So this is very exciting to us. The mice during the study did perfectly fine, and now we've kind of succeeded in the development of an orally available bone anabolic agent. So what comes next? Very recently, we're very excited to license the technology from this project to Radius Health. And that's allows us to continue collaborating with this pharmaceutical company on the further optimization of this compound and ongoing preclinical development for osteoporosis and other disease indications. And really importantly, having this compound, in addition to being in a potential therapeutic direction, this has really boosted the ability of our laboratory to study this pathway, both in bone and in mineral metabolism and in other areas where these kinases play important roles. So with that, I'll stop. I absolutely wanted to acknowledge the members of my laboratory. Here we are getting together in 2020 on Zoom and in 2021 in real life. In particular, I wanted to acknowledge Toshi Sato, Caitlin Strauss, and Chris Castro, who are the people who have been most involved in the efforts that were described here, along with the TSC from the Harrington Discovery Institute and our collaborators at Conifer Point and Radius Health. And again, thank you very much for your attention, and it's really a pleasure to be able to come back for this talk today.